Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our keynote speaker, Dr. Rodney Dietert, Professor Emeritus at Cornell University and author of The Human Superorganism. Dr. Dietert has a vision for a personalized, whole human microbiome-first approach to health, in which managing our microbes is a key to sustainable healthcare. We are very pleased to welcome Dr. Dietert with the keynote address, The Big Picture View of Our Tiny Microbes.
Hello, I'm Rodney Dietert, Professor Emeritus, Cornell University. These are the disclosures. I want to begin by thanking the World Asthma Foundation for the thousands of hours involved with organizing, sponsoring, and delivering this event. Also thank the speakers for their commitment to excellence and for their summit contributions. And thanks to the thousands of registered attendees who range all the way from those afflicted by non-communicable diseases to hundreds of researchers and educators participating as well. And finally, thanks to my wife, Janice Dietert, for her editorial suggestions. The hope is that the vision and information conveyed across these presentations will help to improve lives and guide us towards sustainable healthcare. First, some definitions. The microbiome is a collection of microorganisms and their genes living in a particular environment. So this includes bacteria, archaea, viruses, fungi. And for humans, it can involve the whole human body, the human microbiome, or a specific body part, such as the gut microbiome or the skin microbiome. Microbiota are a group or collection of microbes. So we might talk about the gut microbiota. Holobiont or superorganism is an organism made up of multiple species that operates as a single unit. Dysbiosis is an imbalance within the superorganism and usually wrong microbes or microbes in the wrong location that will promote disease. Pathobiont are microorganisms that promote pathology if placed in the wrong environment. This often occurs with dysbiosis. The issue I'll be addressing is that thus far, Western medicine has operated as if humans are a single species rather than a superorganism or holobiont. Prevention strategies and treatments have been slow to embrace the microbiome, and there's been a tendency to chase the presenting symptoms or treat those symptoms rather than to correct underlying imbalances that occur within physiological systems. Focusing on the present issues rather than integrating across health risk management that expands the lifespan has also been a problem relative to comorbid diseases that arise later in life. So to address this, it's important to discard outdated scientific dogmas from the 20th century that we know are no longer correct and may lead to misguided approaches, and also to apply microbiome first approaches that embrace our majority, and that would be our microbes. They are the majority of our genes, and we need to protect friendly microbes and use rebiosis as a go-to strategy. I'll first describe some microbiome fundamentals, including the way in which I became involved with the microbiome, which was really by accident. I'll talk about the old scientific dogmas that impede our progress to sustainable healthcare, the importance of immune microbiota co-maturation, depletion and destruction of the human microbiome, and how that is connected with diseases, the two categories, chronic non-communicable diseases and infectious diseases, which are involved with microbial colonization resistance. And finally, conclude with issues of safety for the whole human, including the microbiome. I became involved quite by accident with the microbiome because I'd been working on the developing immune system and protecting the immune system of children, and I was challenged to prepare a paper that described the one sign or the one measurement you could make in a newborn baby that would best predict the trajectory that baby would follow in terms of whether the life was filled with health or filled with disease. And while we know that many things, diet and environmental exposures and lifestyle affect health as you age, nevertheless, this was to identify a useful and the best predictor at birth for what might happen later in life. And I thought, well, it surely involves the developing immune system. That's what I'm working on. But I was completely stymied trying to prepare a paper, went to bed very frustrated that night, and awoke in the middle of the night from a very vivid dream with one single idea. And that idea was that the baby needed to self-complete. The baby needed to form the human microbial superorganism and be seeded with the microbes that were intended. And that led to what is called the completed self hypothesis. And my wife helped me to scramble my middle of the night ideas into a paper that described the importance of family sourced microbiota being seeded during birth through vaginal delivery when possible, and that would seed the microbes into the airways, into the GI tract, the skin, your genital tract, and the like. And the importance of this, and being able to measure that, the extent to which that had happened as an indicator of future health and risk of disease. So it turned out that that paper was seen by some British filmmakers, including Tony Harmon, who's a speaker on this summit program. And they allowed me to present this in the film Microbirth. And that led to a variety of speaking opportunities and other papers, including National Academies of Science talk, and the ability to prepare a book, even without a proposal, for the popular press on this topic. And so really the hope has been that coming at the microbiome from this other perspective of working on other things, including the developing immune system, could be useful in terms of some of the ideas that could be applied. So I want to start by describing the fact that Earth as a planet is primarily a microbial planet. That is the predominant life form on Earth. And microbes are distributed across Earth into the harshest environments. We, as a human superorganism, carry our own microbial bubble. So we're accumulating microbes and depositing microbes as we move through hotel rooms and cars and transportation. And we're very good at traveling to different places on Earth and distributing microbes. We're sort of like the Johnny Appleseed of microbes distribution on planet Earth. But the important thing is we need to understand that we are a part of the life that is the predominant life form on Earth. And we have a role to play in planet Earth, and you should never feel ashamed of human contribution to Earth. This is particularly emphasized by the fact that we contain genera of bacteria that are located in the harshest environments on Earth. The Yellowstone Geyser, under glaciers in Antarctica, eating minerals and rocks as food, the Marianas Trench and the extreme pressure at that depth, and the huge salinity of the Great Salt Lake and the Dead Sea. And genera of those microbes are in our gut, as well as microbes that basically are magnetic oriented. They have magnetic features. And it's very interesting because, of course, magnetic fields are one of the ways that birds navigate. And you have to wonder how we might better utilize our own magnetic microbes and bacteria that are present in our gut. So we share genera with life at the extreme ends of Earth. They are at the extreme ends of our gut. And we're a part of those families, that relationship. The Human Genome Project in the late 90s and early 2000s identified the fact that there were 22 to 25,000 chromosomal genes in humans. This was about half of what had been estimated. But that dwarfs in size compared to the almost 10 million genes across the human populations on Earth, with about 3.3 million microbial genes in a given individual. So it is closer to hundreds-fold difference in terms of the microbial genes versus the chromosomal genes. And we're more than 99.9% microbial genetically. When it comes to cells, it used to be thought we were 90% microbial. It's closer to 57%, with some of the most recent estimates of 39 trillion microbial cells versus 30 trillion human mammalian cells. The location of the microbiome is really important. Our microbes are our gatekeepers. They're also the filters for what is outside of our body. And if you are a toxicologist and considered routes of exposure to potentially hazardous chemicals or drugs, then you'd worry about dermal exposure through the skin, oral exposure through ingestion, and inhalation by breathing. Well, these are the locations of the microbiome: skin, GI tract, airways, and urogenital tract as well. So as a toxicologist, I would say, well, the microbiome is the first to see chemicals, and whether they're hazardous or not is really in the perspective of the microbiome. If I were a microbiologist in the microbiology department at Cornell, I am in that department, worried about infectious agents, I talk about portals of entry, how infectious agents enter our body. Well, they enter through breaks in the skin, oral exposure, urogenital exposure, and inhalation. These are the exact locations of the microbiome. Again, the nose, the mouth, skin, urogenital, gastrointestinal system. So these truly are at the critical points for determining safety for our body, controlling elements of that, for determining infectious risk, because they provide protection, first line of defense protection against infectious agents, and can spare our immune system from even having to be involved if we manage the microbes correctly. So the microbes are able to establish an internet. They are basically nature's internet. And this is illustrated by the fact that a plant under attack from aphids is able to communicate its position, its defense, its stress through a soil, mycelial, and bacterial network that will reach another plant and allow a plant at a distance to change gene expression and arm against aphids and be successful in defending against them. So this plant communicates through nature's internet, microbial network, to change what happens and give information to another plant. I can assure you plants are not the only things that can do this. We simply need to figure out how using our microbes can help communicate person to person and through the environment, through these media, using nature's internet to give us the capabilities that we have not yet really recognized. The other point is that we often feel better when we're immersed in nature, when we're in a forest, when we're gardening, and there's a reason for that. And that's because the environmental microbes, such as Mycobacterium vaccae in soil, are able to modulate us, modulate our physiology, and they're also in contact with microbes inside of us and on our skin. So this bacterium, for example, will reduce stress. It will reduce anxiety. It's very useful for PTSD. And it explains why we need these kind of microbes. We need these exposures to stay healthy. And they can actually reduce our dependency on the need for certain medical care and drugs. So as part of the veterinary college at Cornell, I'm almost obligated to show you at least one animal slide. And the point of showing you dogs is that they often do not greet face to face. It's more nose to tail. And the reason for this is they are among the best species at sampling the microbiome and knowing the microbiome through the volatile organic chemicals that the microbes emit. So they can detect these at a level that we're not, that is thousands of fold above our sensitivity. And when they're doing this, they're basically saying, who are you as a canine superorganism, and do you have any useful microbes? And I used to think, oh, this is disgusting behavior by dogs. No, it's brilliant behavior. They're a lot smarter than I used to think. And this is also proven by the capacity to train dogs to detect bacterial pathogens, or very specific bacteria. And this is Cliff the Beagle, who was trained to detect C. difficile, which is a hospital-associated life-threatening infection. And Cliff was able to detect this days before other instruments in individual patients and in hospitals in terms of outbreak. So your dog probably knows where you are in terms of your microbiome. You just haven't asked the dog in the right way. Because the microbiome is at these portals of entry, at these routes of exposure, they are going to react to the chemicals that are in environmental pollutants, in drugs, diet, food, and supplements. And they'll do several things. Some of them will sequester this. They want this. This is what I need. I've been waiting for this. Thank you. And they will hoard it. They may avoid it and exclude it because they do not want it. In fact, they will deliberately not expose themselves to it if they can avoid it. They'll metabolize it. And depending on the metabolism, they'll either make something useful for you, they could make something dangerous for you. It depends on the microbes you have in your microbiome. You can control that, and your healthcare providers can assist. They may signal, so they produce other chemicals that affect gene expression in other microbes and affect gene expression in the human mammalian cells. Some of them may die. They may be sensitive to it. Some of them may grow. It may be the food they've been waiting for. They'll expand in population, or they may relocate. And this is a bad thing. If they translocate, they usually move to an environment where they'll cause more problems than benefits. And so you don't want translocation in general. They need to stay in their wonderful niche, their wonderful habitat, and not be forced to other parts of the body. So individual status, personalized medicine, involves a microbiome as well. And depending on your microbiome, it can make an environmental chemical, such as arsenic in drinking water, more toxic to you, or it can better protect you. And your individual risk for a given single exposure to arsenic in drinking water is determined by your gut microbiome. So you may be more sensitive or less sensitive than your neighbor, depending on your microbiome makeup. It can determine whether a drug has efficacy or toxicity. And there are examples of this. Digoxin, for one, can kill some patients, can be ineffective in some patients, or beautifully effective in some patients, all based on the level of one bacterium in your gut. It can determine the physiological response to certain foods and supplements. Not all of us respond the same way, and understanding your microbiome makeup can help determine your likely response to a particular diet or particular food or a supplement. So we can pair these, we can work these in concert. So the old dogmas that hold us back from healthcare are things, some of which I used to teach when I went to Cornell in the 1970s, and we now know is simply wrong. One of those would be that most microbes are a threat to human health. Well, no, most microbes are actually safe, and many of them are needed by us if we're going to be healthy. For example, some of them make vitamin K. We can't make that ourselves. And we need them to metabolize bile salts. And we need them to digest our food and produce certain things that actually benefit our bodies. So they are required for good health. And there are a few that are pathogens, but they're much outnumbered by those that are safe. The newborn is fully prepared immunologically to fight all diseases. I used to teach that in immunology in the 70s. Not so. The infant coming out of the womb has been specially attuned immunologically to being in the womb. There have been some things that were not allowed to develop and to mature because they would have presented a problem with carrying the fetus to term and the mother's immune system as well. And so the newborn is skewed immunologically at birth, and this has to be corrected. It's corrected by co-maturing with the microbiome. These are communicating with each other, the immune system and the microbiome, and that is required to immunologically balance the infant and the infant being prepared to fight disease. Otherwise, without this and in the absence of this, the infant is born with a predisposition for inflammatory generated disease and infection, and a higher risk of infections. And that's simply the case. It's a programming for disease, if not corrected through microbiome cooperation. So all the more important to do that. Third, we now know that the immune microbiome interaction is so biologically sacred, it can literally cause organisms to separate into new species. And this occurs when the immune system does not like the microbiome in a hybrid created between organisms and kills it. It kills it through massive inflammation, which, by the way, sounds a little like what happens at a lower level with chronic diseases, where you have inappropriate inflammation. So in this case, you can either create species separation, or you can have organisms in the same species completely based on how the immune system and the microbiome like each other. That is a very new finding by Seth Bordenstein at Vanderbilt and other colleagues. Non-communicable diseases are completely not able to be communicated or not infectious. And that's not completely true. It's a very soft boundary there, and we now know there's specific transfers of microbes that can promote specific non-communicable diseases or chronic diseases. And I'll describe some of these. I'll show an example in a review on Staphylococcus aureus and asthma. The human mammal is the target of environmental health risk assessment, and safety is determined by that. But in fact, it's the human holobiont superorganism that is the target, and that's how we need to do safety assessment. That's what toxicological safety has got to mean. So if we don't know if it's safe for the microbiome, we don't know if it's safe. So how does the microbiome affect our health and our longevity? Well, there's a global effect that connects the microbiome to sleep regulation, to the circadian clock, to inflammation and its regulation, and to aging, particularly oxidative damage generated telomere length, because that again is affected by the microbiome and immune interaction. So telomere length can be influenced by the microbiome, and we can have a euthanizing effect by managing our microbes better. So the microbiome can drive what I call, and others previously in literature have defined as the health span, and that is not just how long you live, but how long you are healthy during that lifespan. And that's important to be able to have and extend a healthier period of life. There are also systems biology-focused effects or physiological system effects. I already mentioned the importance of the immune system microbiome interaction, and the microbiome affects not just maturation, but regulation as well. The gut is a second brain, and it makes more neurochemicals in the gut than in the brain, and the microbiome, the gut microbiota, contribute to this significantly. They also regulate gut production of it by mammalian cells. So you can actually affect the levels of neuroactive peptides by managing your microbes in the gut. Microbes regulate the hepatic metabolizing enzymes, so they regulate liver function. They are an important endocrine organ in terms of what they produce. They're important in bile acid metabolism, lipid metabolism, very effective in terms of systemic biology regulation. They regulate the HPA axis, and they produce a set of chemicals that are fatty acids, small fatty acids, that are epigenetic regulators, and we never dreamed these chemicals were doing the kind of things that have now been defined in terms of controlling our own cell regulation. So an area called psychobiotics that John Cryan and his colleagues have defined has been important because we now know different bacteria produce different neuroactive peptides. And you can balance neurochemistry and balance what is affected through 3 different routes to brain status as well by adjusting bacterial ratios and bacteria balance. So this gives a new hope that managing microbes may help reduce the need on drugs that could produce side effects that are used for things like major depressive disorder and the like. And by simply adjusting what's already in the gut, but in terms of different ratios, you can adjust neurochemical balance. The immune system can either be in sync and be fully matured and in homeostasis with the microbiome or not. And I like these 2 images that you might remember for what happens when you have ineffective maturation of the immune system. The microbiome's deficient, not in place, the wrong microbes are there, and you're going to have misregulated inflammation eventually leads to pathology and disease in tissues. Or you can have a nice homeostatic regulation, very effective immune maturation, and everything is beautiful, so to speak. Now, the baby at birth, as I mentioned, is programmed more for disease than health, unless corrected and balanced, rebalanced. And that's because there's a preferential T helper 2 environment in the womb that transfers out to the newborn and has to be adjusted. There are aspects of inflammation that must be ramped down after birth, or that will produce disease. And specialized innate immune populations have to be recruited to the mucosal tissues, to things like the gut and the lung, urogenital tract, and these must be signaled in by the microbes and provide immune homeostasis. So it's important to have these in place to populate these locations that are your filter and gateway. Bifidobacteria play an extremely important role in early postnatal immune maturation, and that much we know at the moment. The other component is feeding the microbes. You seed and you feed. And human breast milk is very important in the infant gut. So breast milk, of course, is the gold standard. It's nature's food for the infant. And nutritionists knew that it contains a major element, human milk oligosaccharides. But the problem was we can't digest those. Our human mammalian cells can't use them. So they're not there for us. They are the food of the microbes. And that tells you how important this seeding event is, how important the microbiome is for the infant. Because the main food supply in the breast milk is not for us, it's for our microbes. The other components are that there are additional microbes seeded from the breast milk. It contains a microbiome that is seeded. And this evolves and changes during the weeks of maturation of the infant. Breast milk changes profile. You have immune hormones or cytokines. You have IgA antibodies that help protect the infant and also modulate the immune system. And you have iron-regulating proteins like lactoferrin. So the baby has the infant gut microbes. There are mucin layers. There's actually a bilayer of thickness of mucin and a one-cell thick gut lining barrier with specialized immune cells that have been recruited underneath. And what we now know immunologically is that over 2/3 of the human immune system is in the gut. We never dreamed that was the case back in the 70s, but we now know that is true. So this is really one of the major regions. If you want to look at human immunology, go to the gut first. But it means also that the different separating potential pathogens from immune cells, potential problem players among these microbes, from here is just this one cell thick layer and the mucin. And I'll tell you why it's so important to consider this as a unit and to ensure that this integrity is maintained and that you don't have immune dysfunction and improper inflammation arise if this is damaged. So as a result, I have advocated supporting a systems biology approach and thinking about the microbes, the mucin, the one cell thick gut lining, and the specialized immune cells as a unit called the microimmunosome. And we need to study each of these individually. That's important in terms of research. But eventually we have to put them together because there's bidirectional communication. And if you affect one of these, you're going to affect the others. So it's important to understand the unit and to manage the unit so that we reduce improper inflammation. We have the right balance of microbes to help do that. Now, Maria Gloria Dominguez-Bello, doctor, faculty member at Rutgers, has been instrumental, along with her colleagues, in defining the microbiome diversity in indigenous populations in South America versus urban areas like Sao Paulo, Brazil, and Western cities, other Western cities. And the fact is that through her work and Martin Blazer's work, we know that we've lost over 1/3 of microbiome diversity in Western countries. And that's part of urbanization, diet, antibiotic use, unsafe things that damage the microbiome that we never knew about. But this is important because Dr. Dominguez-Bello has shown that if you take these people and immerse them in this environment, particularly children can increased diversity of their microbiome. They can become more age-matched, similar to indigenous populations. Adults, it's a little harder. They do change, but their age matching is not as complete. So the earlier, the better. But what we know is these populations don't have obesity, cancer. These do. And you can get there very fast. Lose diversity, increase non-communicable diseases by simply immersing yourself in the urban environment. So we have the clues how to do things differently and more usefully. This is also shown through things like elective cesarean delivery, where if you don't have the seeding properly, if you don't collect mom's microbiomes and install them medically after birth, and Dr. Dominguez-Bello has developed a procedure to do that, then you're going to have altered immune development, altered cytokine production, more hospital-associated microbial seedings, elevated risk of diseases with the mucosal immune system dysfunction. The same thing can happen relative to production animals. The antibiotic use as growth promoters was a devastating thing. It increased antimicrobial resistance, increased zoonotic risk for humans as well. Again, this is the animals were essentially being operated on a depleted microbiome in the end. You can see this with zoo animals. Howler monkeys are an example, where there are 57 plant species that they access in Vietnam as food. If you go to Asian zoos, they usually have fewer plant species, and that can erode the microbiome in part because of diet change. If you go to some Western zoos, there's only one plant species that may be used. And this dramatically reduces microbial diversity. So what happens? Because the spaces in the lung, in the gut, in the urogenital tract, on the skin, they're going to have microbes. It's just a matter of where they come from. Well, interestingly enough, these animals become humanized. They actually acquire microbes from the animal handlers. That's really who they're exposed to. And because they lack microbes, diversity, they will acquire animal handler microbes. The same thing happens among lab animals, such as rats and mice, because they are not as diverse as wild rats and mice in terms of microbiome. So they will actually acquire microbes from the animal handlers, and their microbiome is affected by the breeding facility, caging, bedding, feed, the building they're in. So if you're doing research with lab animals and you change technicians that are handling them, you change the room, or you change the building, you could well affect the physiology of those animals and the results of your experiments. 6 factors led to the microbiome depletion and this current epidemic of chronic diseases. Antibiotic overreach is described beautifully by Martin Blaser in his wonderful book. The food revolution and diet, moving to reduced use of breastfeeding and increased use of processed foods. Urbanization, where we have megacities, we see trees and parks and we think it's wonderful, that we're just good to go, but in fact they are microbial wastelands. And again, you can look at Dr. Dominguez-Bello's work to see this. Birth delivery mode, where there's increased elective cesarean in some countries, and that reduced seeding of babies. Misdirected efforts at human safety, where we never screen for safety for the microbiome, and we're now paying the price. And mammalian-only human medicine, where we need to include the microbiome, we need to go to microbiome-first approaches, and we can no longer treat the human as a single species. So there are 4 basic pillars that pertain to these chronic diseases, or NCDs. They are likely to be programmed in early life, so what you do in pregnancy, in the infancy, in the early childhood is really important, because that's when you set the programming of the physiological systems and can misprogram certain things that will contribute to later life chronic diseases. Uncontrolled inflammation is key to maintaining chronic diseases. So if only we could get to the roots of that, we would have a better chance of effectively treating these conditions and reducing the likelihood of comorbidity. Having one NCD currently increases the chances of developing additional NCDs. And we know, in fact, the spectrum that are likely to occur in a cohort of children who are obese or children with asthma. Microbiome status affects the risk of NCDs and determines also the effectiveness of drug therapies, as I'll show you. So this is an epidemic, and NCDs account for the major cause of death, majority cause of death globally, including developing nations. It's over 70%. And it's estimated by the World Economic Forum, Harvard School of Public Health, that the expense toward NCDs is going to be half of the global GDP by 2030. So we already can't afford this, but we're really coming to a crisis in terms of sustainability of what we're doing in managing chronic diseases. These diseases are emerging at earlier ages, so we have a greater prevalence of pediatric celiac, pediatric arthritis, for example, that used to be later diagnosed conditions. There's reduced, there's loss of life years, increased disability years, and a greater need for caregivers. We're running out of caregivers with some of these conditions. And we now know that earlier in life is better in terms of trying to correct NCDs. Doesn't mean you can't do useful things later, but adolescence is the last outstanding chance to do something. So we need to do things differently in managing these differences. In 2010, a group of us collaborated to publish this Venn diagram, asking the question, what if a cohort had a particular chronic disease, immune-inflicted chronic disease, like childhood asthma, inflammatory bowel, autoimmune thyroiditis, psoriasis, recurrent infant respiratory infections, frequent infections? What was their prognosis relative for that cohort relative to risk of additional chronic diseases? And the answer is there's a predictable group of diseases that they're at a higher risk for than the general population. And you can see these outlined in each of these ovals. What's interesting is at the epicenter of these are three conditions: depression, sleep disorder, and atherosclerosis or heart disease. And these are three of the most prescribed drug conditions that exist. They are shared by many comorbid initial NCDs. So we could really put a dent into requirements of prescription drugs by tearing this apart. Now we know a lot more about comorbidities 12 years later than when we published this paper. So here's the update actually from 2021, published in Biomedicines, with 36 comorbidities for asthma. Some of these you would anticipate, allergic triads such as rhinitis and dermatitis, food allergies, but some not, like depression, obesity, GERD, osteoarthritis, hypertension, type 2 diabetes, lung cancer. So currently, with the current management, this is what's ahead for the cohort of asthmatics. This need not be the case. We can do a better job of protecting against these comorbidities, and this should not be the track forward. If you look at protective factors for asthma in early childhood, this makes a lot of sense microbially. So vaginal birth is one of the tops, because that's seeding with microbes or interruption of seeding with microbes if it's an elective cesarean delivery. Prolonged breastfeeding. Again, you're seeding with more microbes. You're feeding and nurturing the development of the microbiome and the immune system. Growing up on a rural lifestyle, rural farm. So you have exposure to allergens and microbes in the environment and with animals, and that combination leads to a more diversified childhood microbiome. Lack of antibiotic exposure, because antibiotics in the end destroy friendly microbes along with the pathogen. If you don't replenish that, if you don't restore the microbiome, you leave patients wide open for elevated risk of recurrent infections and chronic diseases. High maternal vitamin D and zinc intake, these are very important for a variety of physiological systems, including the immune system, vitamin E intake, healthy diet, Mediterranean diet, and rebalancing the immune system of the infant, including the full spectrum of T helper function, are protective against asthma. In addition, infant bifidobacteria are really important in seeding and feeding events because they help digest the human milk oligosaccharides. If you go to obesity, it's even worse. 43 comorbid NCDs for the cohort diagnosed with obesity. Some of these you'd expect, metabolic syndrome related, type 2 diabetes. Some of them maybe not, atherosclerosis, hearing loss, infertility, Alzheimer's, hypothyroidism, depression, there it is again, the epicenter, 12 different cancers. So again, this is a nasty prognosis for that cohort, and we can improve this. We can do better. One of the ways this is thought to happen is the fact that the conversion from beige fat cells to white cells, fat cells during obesity leads to this becoming an immune organ filled with macrophages that are polarized toward what's called M1 inflammation. And this inflammation actually can spread to other tissues. So it's not that the white fat cells stay in one place and only do one thing. They actually will propagate this immune dysfunction to other tissues, and that may be part of how the comorbidities arise, is through this. So there are, not all species are equal. There are communities of species in the microbiome that do wonderful things together functionally, but there are some examples where single species are really critical. They have critical functions. We can't afford to lose them. So one of these, where there's only one or two species that carry out the function of mucin maintenance, is Akkermansia muciniphylla. And this particular bacterium is critical to maintain not just the mucin layer, but then the integrity of the one-cell thick gut lining. So if you lose Akkermansia, you're in trouble. You're on the way to metabolic syndrome and probable obesity. So there are a number of things that are known to kill Akkermansia. Among these are food emulsifiers. Now, food emulsifiers make your food smooth and keep it from separating, and they're extremely prevalent on grocery store shelves. And they are extremely dangerous because they will destroy this bacteria. So here's an example of something where regulatory agencies may want to take a look at the ramifications of this being in what people think is healthy food, but is not. Heavy metals will do this. Cadmium, it can be, or obesogenic. Some antidepressant drugs and plasticizers, bisphenol A. So bisphenol A, again, is still not banned everywhere. And the question is, why does it take 2 generations of exposure to get a dangerous chemical out of our products? So I think, again, this is a case where here is a keystone species. We need this function, and we know that there are actually food products that are destroying this at the moment. There are other functions that this bacteria carries out besides mucin regulation. It also has pili, like proteins that stick out from it, that modulate macrophage status, and they will protect against the obesogenic polarization to M1. So they move macrophages in the opposite direction. They will block or protect against fibrotic effects in the liver. They will help defend the host against influenza. And that's extremely important, that frontline defense I talked about against pathogens. And they modulate the host serotonin system. So they're modulating neuropeptide active production by other bacteria. Very important. So the old dogma that there's a boundary between infectious or communicable diseases and non-communicable diseases, and that this is absolute and hard, appears to have eroded. We now know that there are some cases where particular pathogens can be transferred and can, in open individuals that don't have a diversified microbiome, produce a particular NCD. And this is now known to occur. And one of these is Staphylococcus A, that has a number of ways to promote asthma, and carriage in infants, in the nasal passages of infants, is a risk factor for asthma because of what Staph A can do. So we need to now think about this boundary being very soft, and that we're ultimately managing microbes. We need to manage and protect against pathogens and pathobionts. And by doing that, by having a good diversified microbiome, that is the frontline protection for both communicable and, in the end, non-communicable diseases. This simply shows an example of where a bacterium, Fragilis in this case, can block an opportunistic pathogen, keep it away from the gut lining, not allow it to kill gut lining cells and damage and make this porous or leaky, and allow immune balance to occur. And what happens if you don't have these friendly bacteria in place? You don't have maximum colonization resistance, and the bad actors can get to the gut lining. And it doesn't take that many friendly microbes to block pathogens. We now understand that very clearly. It's not like you have to wholesale change the microbiome to acquire effective colonization resistance. So how do we know this works? Well, it started by looking at what happened in the poultry industry in the 80s and the 90s, because there was a huge problem, particularly in the Northeast US, where there was a salmonella outbreak in poultry. It was transferred two ways, through the egg and bird to bird, horizontal transmission. And through the egg meant the pathogen was zoonotic for humans. It could infect humans, it could cause illness, and it could kill humans. So food safety was a huge concern, and egg consumption plummeted. And because food producers, farmers are making just pennies on the dozen in their end of the profit, this was devastating. So they needed a quick, effective solution. And the solution they found went back to a Finnish scientist who in the 60s developed something known as the Nurmi concept, named after himself, Nurmi. And that was to achieve colonization resistance by loading the chickens up, feeding them Lactobacillus acidophilus, one of the probiotics that was known back in the 80s. Well, doing this works. Salmonella was blocked. The chickens and the food were safe and safe for human consumption, and it allowed the industry to come back. And this has been used in commercial poultry products ever since. So we have 35 to 40 years of success based on this. You just never hear about how this saved the poultry industry and how we have that amount of data being applied on a pennies profit industry. So does this work? Do probiotics, can they work if you know what you're doing, if you know how to nurture the right probiotics, if you know how to use colonization resistance? Absolutely. Otherwise, you wouldn't have eggs to eat today. So safety. And this is where I was involved directly with a colleague, Dr. Ellen Silbergeld of the Johns Hopkins Bloomberg School of Public Health. And Ellen was on the National Research Council in 1987 when they developed this model that's used to determine human safety, whether it's an environmental chemical or a drug or a food additive. This is the flow chart that's used. So you have exposure to the chemical, you have an internal dose, the biologically effective dose, and you go through to where there's altered physiology or organ function changes, and then clinical disease. And there are a variety of susceptibility markers, age, host genetics, and the like that come into play. And these determine the overall risk of clinical disease. So this is how regulatory agencies use these data to determine if something's safe or not for humans, for human use, for human consumption, for drug administration. But there's a problem. And the problem is what you don't see. And the problem is the gateway, the filter, the frontline defense for humans. And Ellen, being on this committee, said, you know, this has to change. We're not doing safety right. And from my microbiome experience, we had the same thought. So we developed a paper with the simplest figure change imaginable. We put the microbiome in as the gateway or the filter. So exposure through the microbiome gives you presented exposure, because what the microbiome does to the food additive, to the drug, to the environmental chemical determines what happens here. what gets to your mammalian cells, all the way through finally to altered structure, clinical disease. If you don't know this piece, you don't know this. So you can't do proper safety evaluation excluding the microbiome. That's good news, bad news. It means toxicologists have a world of work ahead, work opportunities. It means what we did in the past, not that useful. And this is simply the reality. So we published this in Toxicological Sciences in 2015. So there are examples of drug interactions with the microbiome, where the drug damages the microbiome, or the drug needs the microbiome or an element of it to work. I mentioned digoxin, where one bacterium determines whether that it has a narrow effective range, and in some people it could kill them, or it won't work, or it will work. You adjust this bacterium level, it'll work. NSAIDs, over the counter, used by many, one of the most used drugs in existence, can damage the microbiome. Obviously, dose matters, but if you're using it regularly, it can damage the microbiome. And microbiologists can not only tell you that it's damaged it, they can tell you what NSAID you've been using because of the pattern of the damage, because there's some subtle differences in what gets killed and what doesn't between the NSAID categories. Proton pump inhibitors, maybe you're worried about gastric ulcers from NSAIDs, you take these, they damage a different part of the microbiome. SSRI antidepressants, they restructure the gut microbiome, causing a loss of some needed species. Oral steroids, metformin. Laxatives, antihistamines, on down to cancer therapeutics, where these are needed to be metabolized by the microbiome to be effective. So we now know if only we manage the microbiome, we could increase the effectiveness of cancer therapeutics. That would be a glorious thing. So I want to point out that it's the combination of maternal status, OBGYNs, you can positively affect 2 generations by affecting the maternal microbiome and making sure it's healthy. It's antibiotics, delivery, seeding, it's formula feeding versus breastfeeding, it's exposure to things like heavy metals coming through baby products, contaminants, bisphenol A, food additives. All of these go together in terms of infant status, childhood status of the microbiome, and the physiological systems. So the take-home points are, at birth, the newborn's immune system is skewed toward inflammatory disease. It must co-mature with the microbiome to become balanced. These systems, immune system and the microbiota, grow together. If the microbes are missing, infection and immune-inflicted chronic diseases happen. Nothing should be considered safe for humans unless it's proven to be safe for the whole human, and that includes our friendly microbes. How we produce and consume food, use chemicals, use drugs, shapes our global microbiomes, composition, nutrients, prebiotics, probiotics, and they help drive our own microbiome. So we can manage our microbes, manage our diet, manage our other chemical drug exposures in a way that really helps us be healthier. Healthcare should be practiced using personalized whole human microbiome first approaches. That is really direction to reduce the NCD epidemic, to provide better resistance against infectious diseases inherently and on a personalized basis. It really is all about managing microbes, and that's the key to sustainable healthcare. I want to end by saying that there are 2 of the most prominent scientists involved with destroying dangerous microbes, loved microbes. The first, Metchnikoff from the early 1900s, who was an immunologist that discovered how immune cells, particularly macrophages, kill bacteria. But Metchnikoff wrote a book on probiotics. He believed that you needed to consume fermented foods to be healthier and to have better longevity. And he consumed Lactobacillus bulgaris and believed this was really important for health. So he loved microbes. Alexander Fleming, the discoverer of penicillin, loved microbes so much that he used coloriform bacteria that had different colors to create art as one of his hobbies. So this is an example of bacterial art created by one of the great destroyers of pathogenic bacteria, Alexander Fleming. They loved their microbes. I want to thank you. I want to thank the World Asthma Foundation for sponsorship, for providing this. I want to thank all the professionals who have made presentations. And the hope is that you can use the science-based resources that are available through this summit, and you can access from the convenience of your home and office, and that can help you personally, and it can help drive us towards sustainable healthcare. Thank you so much.